Biochemical ratchets are molecular machines in cells that harness chemical gradients to produce directional motion, enabling critical biological processes like intracellular transport and energy conversion. These molecular systems operate similarly to a ratchet mechanism in mechanics, where random thermal motion is rectified into consistent directional movement using chemical energy, often derived from ATP hydrolysis or other reactions. For example, motor proteins like kinesin and dynein walk along microtubules, transporting cargo like vesicles and organelles within the cell. They achieve this by coupling ATP binding and hydrolysis to a cycle of conformational changes, converting random thermal fluctuations into forward motion. Imagine a wind-up toy that only moves forward even if it jiggles randomly. The energy source ensures its motion is productive. Biochemical ratchets are essential for maintaining cellular organization, enabling communication, and driving processes such as cell division and muscle contraction, illustrating nature's ingenious use of chemistry to create molecular machines.
Biochemical ratchets are molecular machines that exploit the inherent randomness of thermal motion, Brownian motion, and rectify it into directional work using energy input, often derived from chemical gradients or ATP hydrolysis. These machines operate at the nanoscale, where thermal fluctuations dominate, and rely on asymmetrical energy landscapes to bias motion. They are crucial for fundamental cellular processes like molecular transport, force generation, and energy conversion. Importantly, these systems work far from thermodynamic equilibrium, a key characteristic of living organisms that distinguishes them from inanimate systems. Example: molecular motors such as kinesin and dynein are classic examples of biochemical ratchets, efficiently converting chemical energy into mechanical work to transport organelles within cells. Analogy: a biochemical ratchet functions like a one-way escalator, enabling movement in a specific direction by preventing backward sliding through energy gating mechanisms. This analogy helps in understanding their directional control. Chemical gradients represent a form of stored potential energy in cells, arising from unequal distributions of ions or molecules across membranes or within compartments. These gradients are maintained by active transport processes, such as proton pumps, which use energy from ATP hydrolysis or electron transfer. Molecular machines harness the energy stored in these gradients to perform mechanical work, such as pumping ions, moving cargo, or synthesizing ATP. The ability to couple the dissipation of these gradients with mechanical processes is a hallmark of biochemical ratchets. Example: in mitochondria, the proton gradient generated by the electron transport chain drives protons back into the matrix through F1-FO ATP synthase, which converts this flow into the mechanical rotation needed to produce ATP. Analogy: a chemical gradient is like a reservoir behind a dam. The potential energy of the water is harnessed as it flows through turbines, just as proton flow powers molecular machines. This analogy illustrates how energy is converted and utilized. Brownian ratchets take advantage of random thermal motion and combine it with an asymmetrical energy potential to produce unidirectional movement. These systems rely on periodic energy input, such as ATP hydrolysis, to overcome energy barriers and rectify motion. By synchronizing energy release with molecular fluctuations, they effectively trap progress in one direction, ensuring a net bias despite chaotic motion. This principle is particularly effective in environments where thermal noise is unavoidable, such as the crowded intracellular milieu. Example: myosin motors interacting with actin filaments use a Brownian ratchet mechanism to rectify motion. This allows for efficient muscle contraction and cellular movement. Kinesin and dynein are motor proteins that walk along microtubules, transporting vesicles, organelles, and other cargo within the cell. Their movement relies on a hand-over-hand stepping mechanism, driven by ATP binding, hydrolysis, and release. The energy released during ATP hydrolysis induces conformational changes that move the motor protein forward while ensuring tight coupling to the microtubule, preventing backward diffusion. These motors are highly processive, taking hundreds of steps without detaching from their tracks, and their directionality is dictated by the polarity of the microtubule. Example: kinesin transports vesicles containing neurotransmitters from the neuronal cell body to synaptic terminals, enabling rapid and efficient communication across neurons. Analogy: kinesin and dynein are like delivery trucks navigating one-way roads in a city, each truck specialized for movement toward specific destinations. Scientific explanation: F1-ATP synthase is a rotary enzyme embedded in the inner mitochondrial membrane that synthesizes ATP. Using the proton gradient generated by the electron transport chain, protons flow through the FO subunit, driving the rotation of a central shaft, Ga subunit. This rotation causes conformational changes in the F1 catalytic subunits, allowing them to bind ADP and Pi and catalyze their conversion into ATP. The entire process is highly efficient, converting the energy stored in the proton motive force into chemical energy that powers nearly all cellular activities. Example: in a single second, a single molecule of F1-FO-ATP synthase can produce up to 100 ATP molecules, highlighting its efficiency and speed. Analogy: F1-FO-ATP synthase works like a water wheel, where the flow of water, protons, turns the wheel, generating energy to power a mill, ATP synthesis. Scientific explanation: DNA helicases are specialized enzymes that unwind double-stranded DNA to facilitate replication, repair, and transcription. These molecular machines move unidirectionally along the DNA strand by coupling ATP hydrolysis to conformational changes, allowing them to disrupt hydrogen bonds between base pairs. The ratchet mechanism ensures forward movement by stabilizing the enzyme on the DNA backbone while preventing backward sliding. DNA helicases are highly efficient, unwinding thousands of base pairs in a single binding event. Example: the bacterial helicase DNAB unwinds DNA at the replication fork, creating single-stranded templates for DNA polymerase to synthesize new strands. Analogy: a DNA helicase functions like a zipper that is actively pulled apart, using energy to separate the strands of a rope, DNA, for replication. Muscle contraction is driven by the cyclic interaction of myosin motors with actin filaments. Each cycle involves ATP binding to myosin, triggering detachment from actin. ATP hydrolysis then resets the myosin head, allowing it to reattach further along the filament. A subsequent power stroke generates force as the myosin head pulls the actin filament. The coordinated action of billions of myosin-actin cycles produces the macroscopic contractions required for movement. Example: cardiac muscle contraction relies on the synchronized action of actin-myosin ratchets, generating the force needed to pump blood throughout the body. Peristaltic pumps are molecular machines that transport substrates through sequential cycles of conformational changes, mimicking the rhythmic contractions of biological systems like the gastrointestinal tract. In cells, these pumps are powered by ATP or ion gradients, and their motion is precisely regulated to ensure unidirectional transport. This mechanism is common in secretion systems and protein translocases. Example: the bacterial Sec-Y-egg translocon uses ATP-driven cycles of conformational changes to push unfolded proteins through the membrane, ensuring proper export or insertion. Analogy: peristaltic pumps are like squeezing toothpaste from a tube, where rhythmic pressure ensures continuous forward movement without backward leakage. Artificial biochemical ratchets are engineered molecular systems designed to mimic the functionality of natural molecular machines. These systems often use DNA origami, synthetic peptides, or programmable nanostructures powered by chemical energy, light, or electric fields. Such systems have potential applications in targeted drug delivery, nanorobotics, and biosensing, with researchers working to optimize their efficiency, stability, and programmability. Example: synthetic DNA walkers have been developed to move along predefined tracks using chemical gradients, demonstrating proof of concept for programmable nanomachines. Analogy: artificial biochemical ratchets are like programmable robotic arms on an assembly line designed to perform precise tasks at the nanoscale,